webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website, autism.org.
Now, before we get started, I'll introduce our speaker. Dr. Greg Wallace is an associate professor in the Department of Speech, Language, and Hearing Sciences at the George Washington University. His research focuses on neuropsychological and structural brain development in autism and other neurodevelopmental disorders across the lifespan and their impacts on real-world outcomes. And now I will turn this over to Dr. Wallace.
Thank you so much, Denise. Hello, everyone. My name is Greg Wallace, and today I'll be providing information on eating-related behaviors and their cognitive, neural, and health correlates in autism spectrum disorder. The current presentation is split into 5 parts, including answering the question, why should we study eating in autism spectrum disorder, which henceforth I'll refer to as ASD. So why should we study eating in ASD in the first place? We'll briefly examine what we know about food neophobia and picky eating, or so-called picky eating, or selective eating in ASD. We'll take a look at the understudied area of eating in the absence of hunger in ASD. Tackle the possibility of a novel combination of these 2 types of eating-related behaviors, which are referred to as selective overeating in ASD. And finally, we'll briefly investigate neural correlates of eating-related behaviors in ASD. So, first, we'll start with the basic question, why study eating in ASD or autism? So I would suggest the place to start when answering this question is the fact that suboptimal health outcomes are common in ASD. Thus, for example, one meta-analysis that included more than 1 million individuals with ASD across the lifespan documented an increased risk for developing obesity compared to general population expectations. Furthermore, numerous studies have documented a plethora of other health conditions or risks that are found more commonly among autistic individuals than in the general population, including high cholesterol, high blood pressure, diabetes, etc. Additionally, there's some evidence, albeit often mixed, for decreased particular macronutrient intake among individuals with ASD. And although exercise is obviously important for optimal physical and mental health, models of weight loss in the general population demonstrate that diet exerts a stronger influence than exercise alone. Nevertheless, thus far, exercise and its effects on weight or body mass index or BMI in ASD and in other developmental disabilities has been the primary focus of research, particularly as it informs how exercise and physical activity contribute to suboptimal physical health outcomes. Therefore, we should also evaluate appetitive traits or eating behaviors as possible influential factors in these physical health outcomes in ASD. Two such appetitive traits are one's food repertoire, as encapsulated by food neophobia, for example, and one's drive to eat, such as eating in the absence of hunger. These traits vary considerably in the general population, but what do we know about them in autism? So this takes us to the next section of the talk, wherein we focus on food neophobia and so-called picky or selective eating in ASD. So I'll apologize in advance for the numerous overlapping terms, but I thought it was important to introduce them to you as they are often used interchangeably or somewhat interchangeably. And this field has lots of terms that can be a little confusing at times. So many individuals with ASD are described as so-called picky eaters. Or they're said to have food selectivity or are selective eaters. These traits are often exhibited through food refusal, for example. Somewhere in the range of 60 to 75% of children with ASD exhibit some degree of selective eating. And while most neurotypical children outgrow selective eating at an early age, in many adolescents and adults with ASD, these selective eating behaviors persist. One such eating-related behavior is food neophobia, which I will now discuss further. Food neophobia is indeed the fear of trying new foods. This food neophobia is a common and seemingly adaptive feature of early eating in all children. Not to put too fine a point on it, but just to clarify for the sake of distinctions, once food is in your mouth, neophobia has been overcome, and any subsequent rejection would be considered picky eating. Again, food neophobia typically dissipates and wanes across child development. However, when it is persistent, food neophobia can be stressful and interfere with everyday functioning for the person experiencing it. So what do studies reveal about the presence of food neophobia among autistic individuals? In one of our studies, we found that 8 to 10-year-olds with ASD were much more likely to be rated as food neophobic than their same age peers in the general population. In fact, half of the 8 to 10-year-olds were rated as, of the autistic 8 to 10-year-olds were rated as having food neophobia, compared to about a quarter of their same age peers. Extending these findings upward in age, we also found that autistic adolescents and adults rated themselves as experiencing food neophobia much more than neurotypical adolescents and young adults. So these patterns of selective eating are commonly documented among autistic youth. However, the driving forces behind them are less well understood. There are numerous candidates likely to contribute to selective eating in ASD, but two likely culprits, as they are symptoms of ASD, are sensory processing differences and behavioral and cognitive inflexibility. Therefore, we completed a study to examine the possible relationship between picky eating or food selectivity and both sensory processing, which is based on ratings of oral texture sensitivities and smell sensitivities, as well as behavioral or cognitive inflexibility among 4 groups of participants. These participant groups included not only autistic youth, but also neurotypical youth, neurotypical undergraduate students, as well as youth with anxiety disorders or obsessive compulsive disorder. What we found was that for all 4 groups, not only sensory, specifically oral texture sensitivities predicted picky or selective eating, but also behavioral and cognitive inflexibility predicted picky or selective eating. Thus, both of these classes of behavior played a role. And interestingly, there was no ASD-specific pattern of results. But because autistic individuals have divergent sensory experiences and more challenges with behavioral and cognitive inflexibility, they are likely to be particularly susceptible to developing food selectivity or becoming picky eaters. So thus far, in the little bit of information I've shared so far, I've been describing and discussing selective eating more broadly without reference to its clinical significance. Thus, I just think it's important to very briefly point out that starting with the Diagnostic and Statistical Manual 5th edition in 2013, or the DSM-5, a new diagnostic category was introduced: avoidant restrictive food intake disorder, or ARFID. One could conceptualize ARFID as a more extreme form of picky eating or food selectivity, which leads to clinically significant limitations in food repertoire, nutrition, and growth, and that can also interfere with psychosocial functioning. So I just wanted to flag this for the audience before leaving this topic. So in summary, taken together, what I want you to take from this section of the talk is that selective eating and food neophobia are more common in autistic individuals than in their same age neurotypical peers across childhood, adolescence, and young adulthood. Thus, evidence to date suggests a life course persistence in selective eating in ASD, despite interventions trying to increase food repertoire, particularly for children. Furthermore, behavioral inflexibility and sensory sensitivities are likely key contributors to food selectivity, and thus represent potential targets around which interventions can be, or in some cases have been developed. Additionally, selective eating and food neophobia likely raise the risk for nutritional deficiencies and subsequent ARFID diagnoses, though much more work is needed in this area. Just as a tease, I'd say that our own preliminary data suggests that a large minority, somewhere in the order of about 35 to 40% of young adults, particularly women with ASD, screen positive for ARFID. So I'm going to now turn away from these food avoidance type behaviors and towards food approach behaviors. So I'm going to briefly discuss what we know about eating in the absence of hunger in ASD. So eating in the absence of hunger is, as the name implies, eating foods, particularly those that are highly palatable, when one is no longer experiencing hunger. Eating in the absence of hunger has rarely been examined in ASD, though 3 small studies from 2 samples suggest increased so-called overeating in ASD during childhood and adulthood. Therefore, we endeavored to more closely examine eating in the absence of hunger and its correlates in children and adolescents with ASD across a couple of different samples. Here, the first study, or study 1, included 99 autistic adolescents and 59 neurotypical adolescents who had their height, weight, and blood pressure assessed during a study visit, and whose parents were asked about their child's eating in the absence of hunger, using an item from the child behavior checklist, and about their child's everyday executive functioning challenges, using 2 subscales from the Behavior Rating Inventory of Executive Function, or the BRIEF. First, we found that parents of adolescents with autism were more likely to rate their children as engaging in eating in the absence of hunger than parents of neurotypical adolescents. Second, adolescents with ASD who sometimes or often engaged in eating in the absence of hunger were more likely to meet CDC criteria for obesity than adolescents with ASD who did not engage in eating in the absence of hunger. Finally, adolescents with ASD who sometimes or often engaged in eating in the absence of hunger were more likely to be hypertensive, or in other words, have elevated blood pressure levels relative to adolescents with ASD who did not engage in eating in the absence of hunger. Additionally, adolescents with ASD who sometimes or frequently engaged in eating in the absence of hunger were rated as being more inflexible in their everyday behavior and thinking, such as experiencing difficulties transitioning between activities or physical spaces, than adolescents with ASD who did not engage in eating in the absence of hunger. In contrast, no differences in everyday difficulties with inhibiting behaviors were observed based on eating in the absence of hunger levels among autistic adolescents. In a separate and subsequent online study that included both children and adolescents, a more comprehensive parent rating of eating in the absence of hunger behaviors, the Yale Food Addiction Scale for children, was utilized. In this study, parents reported their child's current height and weight, as well as any challenges with behavioral inflexibility based on the flexibility scale. Just as in the previous study I described, children with ASD were rated as engaging in greater eating in the absence of hunger behaviors than their same age neurotypical peers. In order to examine links between eating in the absence of hunger behaviors and body mass index, or BMI, in autistic youth, we treated BMI as the dependent variable, and then we entered control variables in step 1 of the regression. Eating in the absence of hunger was entered as our primary independent variable in step 2 to examine its unique association with BMI above and beyond the control variables of age, sex assigned at birth, and both anxiety and depression symptoms rated by parents. These regression analyses that revealed that even after accounting for the effects of the control variables, including age, which was positively correlated with body mass index, and anxiety symptoms, which were negatively associated with body mass index, Eating in the absence of hunger was significantly positively associated with BMI in autistic youth. In a separate regression analysis, we treated eating in the absence of hunger as the dependent variable. And then we entered the same control variables in step 1 with behavioral inflexibility, specifically the autistic youth's proclivity to adhere to routines and rituals is our primary independent variable in step 2. Even after accounting for the effects of these control variables, including depression symptoms, which were positively associated with eating in the absence of hunger, inflexible adherence to routines and rituals was significantly positively associated with eating in the absence of hunger among autistic youth. So for this section, I will just say that combining findings from these studies, clear patterns emerge. First, eating in the absence of hunger is more prevalent in children and adolescents with ASD than in neurotypical children and adolescents. Second, eating in the absence of hunger is positively associated with BMI in children and adolescents with ASD. Third, eating in the absence of hunger is positively correlated with behavioral inflexibility in children and adolescents with ASD. But we really have to be careful here and mention caveats. These studies represent just an initial snapshot of behavioral differences and correlates reported for children and adolescents with ASD. We particularly need large-scale longitudinal studies to understand directionality in these relationships we've just observed in cross-sectional studies, which are just one point in time. So in other words, what is the cause? What is the effect? And there might not be a cause-effect relationship between these variables. Also, what other factors might play a role in the undoubtedly complicated interplay between behavior and health-related metrics? Even though I didn't describe it here, we did take into account other factors, such as certain psychotropic medications, so for example, antipsychotics, which when you use some antipsychotics, they can, in some cases, lead to changes in appetite and subsequent weight gain. Another really interesting research direction to consider is the potential self-stimming function that eating in the absence of hunger might serve. We find, for example, in another study that I don't have time to describe here today, that emotional overeating, in other words, eating beyond satiation in the context of stress, is particularly elevated among girls with ASD. Moreover, several anecdotal reports by women with ASD indicate that the oral motor processes around and during eating are more socially acceptable forms of self-stimming than many other forms of fine and gross motor stimming. Thus, they report quote-unquote overeating for this reason, which is fascinating to think about and shows the value of the lived experience in informing research. Okay, so next I want to very briefly consider the possibility that individuals with ASD might be particularly likely to exhibit a subtype of eating-related behaviors in which both selective eating and eating in the absence of hunger are present and co-occur. The reason this is particularly compelling and important is that traditionally, food avoidant behaviors like picky eating and food approach behaviors like eating in the absence of hunger are considered to occur on opposite ends of the spectrum. The co-occurrence or combination of these eating behaviors in autism would challenge that notion. As always, this research question was not developed in a vacuum. We have encountered numerous clinical and anecdotal reports describing the co-occurrence of these eating-related behaviors in ASD, which can be described as selective overeating. Nevertheless, has not been described previously in the research literature to our knowledge. To examine this empirically, we utilize the data collected in the online study I just previously described to you. Again, parent ratings on the Yale Food Addiction Scale for children were used to assess eating in the absence of hunger, and both selective eating and dietary variety or food repertoire were assessed using the Eating Habits Survey. Using the scores from these scales, we could then categorize individual children and adolescents into one of four groups. The quote-unquote typical eating group, the selective eating only group, the eating in the absence of hunger only group, and the selective overeating group. Analyses revealed differences in the proportion of autistic and neurotypical children who fell into these four categories. More specifically, a greater proportion of autistic children than neurotypical children were linked with selective eating only, which was not quite a 2-to-1 ratio of autistic to neurotypical youth in the selective eating only group, and even more so in the selective overeating group. There was a more than 5-to-1 ratio of autistic to neurotypical youth in this group. Additionally, and interestingly, among children with ASD, those who were described as eating in the absence of hunger had a significantly increased rate of selective eating as well, compared to children who were not rated as exhibiting eating in the absence of hunger. Subsequently, we wanted to know if the food repertoire or dietary variety differed for these eating subtypes among autistic youth. The short answer is yes, and particularly so for consumption and willingness to try vegetables, which was lowest, even though the axis is reversed here, for the selective eating and selective overeating groups. And finally, converging with prior findings I described earlier, we observed increased behavioral inflexibility, particularly adherence to routines and rituals, to be most elevated for children who were described as engaging in selective overeating. Now I'm going to turn to the final section of my talk, which will focus on a study that focus on a study that examined neural correlates of eating-related behaviors among autistic adolescents and young adults. I particularly want to give a shout out to Dr. Jason Avery, who completed these analyses and created many of the slides and figures that follow. As I previously mentioned, and you undoubtedly already knew, individuals with ASD have divergent sensory perceptual experiences across modalities, such as vision and hearing, as well as in valence, such as experiencing hypersensitivity to auditory stimuli, but hyposensitivity to tactile or touch-related stimuli. Thus, one prime sensory-related experience to consider when studying, let's say, selective eating in ASD is taste perception. There have been a few studies of taste perception in ASD completed to date. For example, one study, one of the earliest, by Luisa Bonetto and colleagues, examined taste perception among 21 children with ASD compared to 27 neurotypical children. They found no group differences in taste detection thresholds using electrogustometry, as depicted in the photo on the left. However, they found that children with ASD were less accurate in identifying specifically citric acid or sour flavors and quinine or bitter flavors than neurotypical children, as depicted in the photo on the right. At least one subsequent study replicated these findings by showing that autistic adults were less accurate in identifying bitter and sour flavors in a taste identification task. The combination of intact taste detection but impaired taste identification in ASD suggests that low-level processing is intact. In other words, the taste signals are being received by the brain. However, top-down processing and integration of taste information appears more atypical, which is suggestive of a cortical issue or a higher level brain-based issue. Nevertheless, prior to the following study that I'm going to describe, there had been no research examining the neural correlates of taste perception in ASD. So I promise I'll make this quick and easy. The central reception of taste depends upon the cortical taste pathway. It begins with taste receptors in the tongue, which are specialized to detect one of the 5 or more basic tastes: sweet, sour, bitter, salty, and umami. These receptors are connected to taste afferent neurons, which eventually travel directly to the gustatory nucleus of the thalamus and onto their primary cortical representation in the dorsal insula, which is depicted up here in this cross section of the brain. One way for us to assess this cortical pathway is functional neuroimaging, specifically functional magnetic resonance imaging, or fMRI. fMRI, as many of you probably know, utilizes changes in blood flow to different parts of the brain when an individual is presented with a stimulus to gauge which brain regions are linked to different behaviors. Previous neuroimaging studies have demonstrated that both interior and middle regions of the insula are selectively responsive to a sweet tastant. When I say tastant, it's what it sounds like, something you're tasting. And in this case, the sweet tastant was sugary water, which was delivered to an individual while they underwent fMRI scanning. So that's what you see here on the left. This is the contrast of brain activity to the sweet tastant compared to a neutral tastant, meaning a tastant with no taste, no flavor. And these are the brain regions that were ramped up to the sweet tastant compared to the neutral tastant. Thus, we can harness fMRI to answer a couple of basic questions. First, do individuals with AXD experience or exhibit an atypical brain response to sweet tastants, again, sugary water, within gustatory cortex? That gustatory cortex is the insula, and that's the region that was in green in the figure I showed you before. You'll see other pictures of it. Second, in a related but distinct question, is the brain response to tastants, particularly sweet tastants, related to how these individuals rate their own taste reactivity that they experience in everyday situations and in everyday life? To address these questions, we recruited a group of older adolescent and young adult males with ASD, as well as an age-matched group of neurotypical males to complete the study. During their visit, participants completed a number of behavioral assessments and performed fMRI tasks to identify the neural response to tastants and to viewing food-related stimuli. For the gustatory mapping task, this is how we assessed response to the tastants. We used a pneumatically driven magnetic resonance compatible gustometer. That's a lot. So this right here with these tubes and wiring, what this means is that none of this had metal in it, because this is a giant magnet and it has to be able to be used next to this magnet. If it had metal in it, it would be slammed up against the magnet, which of course is hugely problematic. This is the person who's receiving the tastant laying inside the MRI scanner. So this gustometer was developed by John Ingeholm in the Laboratory of Brain and Cognition at the National Institute of Mental Health, where the study was completed. So during this task, what this gustometer is doing is delivering solutions of either sweet sugary water or a neutral tastant directly onto the subject's tongue during fMRI scanning. Thus, we can compare brain responses to the sweet versus the neutral tastants within the insula, or what we're also calling gustatory cortex. In other words, in parts of the insula shown to be responsive to food-related stimuli. When we did so, we observed no group differences, sorry. I'm sorry, I'm struggling. When we did so, we observed no group differences between the autistic and neurotypical adolescents in how much their insula ramped up when presented with the sugary water compared to the neutral tasting solution. So there were no differences in the gustatory cortex response to tastings. Turning to our second research question, we examined the relationship between sweet versus neutral tastant brain response. So that's expressed as a % signal change in the brain response. And how that related to self-ratings. So that's what the y-axis is. The x-axis on these is self-ratings of taste reactivity in everyday life. What we find here is fascinating. There is no association between self-rated taste reactivity and the brain response to the sugary water solution in the neurotypical group. However, among the autistic adolescents and young adults, there was a strong positive relationship such that those with greater taste reactivity in everyday life had greater brain response to the sugary water relative to the neutral solution. So, based on this study, we find that although there is no evidence of overall atypical gustatory cortical function in autism, activity in gustatory cortex is modulated by individual differences in self-rated taste reactivity. This suggests that atypical brain function for individuals with both autism and food selectivity could impact BMI and various health sequelae via dietary variety. Keep in mind that the participants in this fMRI study were not selected based on their eating behaviors, only on their autism diagnosis. Thus, a study which focused on those experiencing selective eating might have provided different results. As you could probably guess, I strongly suggest we need more studies of cognitive and neural underpinnings of eating-related behaviors to better inform intervention and etiology of eating differences in autism across the lifespan. Overall, although we've known about eating-related differences in autism since some of its first descriptions in the research literature in the 1940s, our knowledge base is surprisingly small. More work is needed, including establishing longitudinal or closer to causal relationships between eating-related behaviors and both what precedes or predicts them, such as sensory, cognitive, and neural factors, as well as what follows them, including physical health outcomes. More intervention work is needed, both for food avoidance and food-seeking behaviors, to improve physical health and overall quality of life in ASD. I also want to clearly acknowledge other limitations of some of the work we have completed to date and that I described here, including, for example, reliance on body mass index or BMI as a health indicator in a couple of studies described here. BMI, as we all know, is a blunt metric at best. We need much more refined and meaningful outcome measures, such as well-validated physical health condition diagnoses that stakeholders, particularly autistic individuals, deem important in order to draw more definitive conclusions about links between eating-related behavior and critical health outcomes. I'd like to acknowledge the families and autistic individuals who participated in these studies, as well as my many collaborators and funders for making this research possible. And for more information, here's a link to my lab website and an email address. I can now turn to the Q&A if that's okay, Denise.
Sounds great. There are a lot of questions.
Okay. All right, so I will just start at the top and see what I can and can't answer. I'll do my best. So the first question asks, and I'm sorry, Denise, do I do the Q&A or the chat? I'm only looking in the Q&A right now.
Yes, if you could look at the Q&A, that would be great. And if people
Okay.
type their questions into the Q&A section, that would be great.
Okay, so the first question says, sometimes a certain food, fruit, vegetable can cause GI discomfort and even pain. While ASD groups have difficulty describing or even pointing out or recognizing the issue, their only choice would be avoiding that type of food. How can we distinguish these factors? Oof, this is a very difficult question to satisfactorily address. I think in the end, this comes down to a lot of communication related issues and other things like interoception, understanding how our body signals, understanding how to understand our body signals and what might be creating the signals that can be difficult to understand. I don't have a straightforward response to this, I'm afraid, but it's a really important question to ask and try to answer. And because most of my work has been on the eating side, not the GI side, I would suggest you talk more to folks who have focused on these kinds of things, which are incredibly important. So if you can't find answers, you're welcome to email me and I can try to find either someone else to consult or something else. So let me just keep going because I could ramble and get to no questions. The next one says, how can we introduce vegetables and fruits to our kids with ASD? This is a great question that you get a lot of. And the short answer is it's a tricky one, right? Which is, I think the biggest challenge that parents of all kids, not just autistic kids, face when they're introducing a new food is that they essentially give up too soon. This does not mean, let's be very clear, this does not mean that you force your child to eat something, right? I'm not condoning any of that. But introducing a food, it actually takes a lot more presentations of a food for a child to become comfortable with it and be willing to try it or explore it. And try it doesn't even mean like eating it and swallowing it. It can even mean just interacting with the food in some way. So there's a lot of work around, because again, picky eating is very common in all children when they're really young. So I think the first thing is figuring out a way to introduce it that is not met with immediate disdain or dislike, and it's just tolerating it, even if it means just tolerating it in a plate on the side, not necessarily again putting it on in their mouth or to their lip or tasting it or anything like that. So it has to be, you have to be patient, and it's and people are talking about food chaining as well, those kinds of things. So I don't have an answer to the next question about the effects of prebiotic, probiotic. I don't feel like I have the expertise to answer that question. It's an interesting question, but I don't have the expertise. I don't know of a study that could answer that, but I'm happy to try to look that up if you can't find anything. Another person asked, how is avoidance of any food of one particular color explained? Well, food avoidance, what I described study-wise was a group-based study. And the two areas the two sensory factors we looked at were oral texture, so literally the way something feels in your mouth, which if we all think about our own food likes and dislikes, that is the number one thing that leads you to dislike a food. So for example, a lot of people dislike things like yogurt or things like eggplants or grapes or tomatoes. And it's partly because you're encountering these textures, like in the case of yogurt, or something that's lumpy. People hate that. Or when it comes to things like eggplant or grape or tomato, one explanation is that you have something that has some solidity on the outside, you bite into it and it's all squishy on the inside, it's unexpected, people just don't like it and they have a visceral response to it. So we find that oral texture is the number one on a group level predictor, but there are plenty of individual descriptions and cases of people who do not like foods based on color, based on temperature, based on other kinds of sensory features that aren't related to the ones I talked about. So again, chaining and other things could be an issue here that could be explored. And it could be because that color, it might not be that particular food, it might have been a food of another color that turned your child off, and they associate that color with that other food, and it's a no-brainer, because we are really fast to condition ourselves when we eat. Again, if I think about having had food poisoning, and the food I had food poisoning, it took me years to have, well, sorry, let me take that back. The meal I had after having food poisoning, even though it's not what made me sick, it took me years to eat that meal again, because your body is so sensitive and so adaptive in a good way, because it's perceiving that as dangerous and having upset, well, just having made you ill. So again, a person asks about cold food. It's the same kind of thing as above. There's all kinds of sensory, that's the temperature thing I was just talking about. And that could be tooth sensitivity, sure, but it also could just be a temperature related issue. So you'd have to discern that based on, well, you could probably do an experiment of some sort. And this is the kind of thing I'd even talk to my pediatrician or others about. Or if you have an OT, for example, that might be even better. How to differentiate if a kid is eating absent or really getting hungry every 1.5 hours. Well, eating in the absence of hunger, this is a tricky one, right? Because eating in the absence of hunger, there needs to be some insight. The thing is, I think just the modern lifestyle sort of, I don't want to say promotes, but makes it easy to eat in the absence of hunger. So in other words, sitting in front of the TV, sitting in front of any kind of electronic screen of some sort, it's easy to just eat without, you're not thinking about what you're doing, which is why a lot of people's interventions around this are about mindfulness. And even, there's really popular dieting, I'm trying to think, what's it called, Noom, other kinds of systems. They focus a lot on using different terms, but a lot on being mindful when you're eating. So that's really important this is like working with your child to help them understand their bodies and when they feel hungry, what hungry feels like to them, and then what that means in terms of their eating. Okay, I'm trying to see if there's any, how can I, oh, I wonder why physical activity is not in the model is a question. And the short answer is, maybe you saw in one of my earlier slides that actually physical activity and exercise are incredibly important when thinking about physical health outcomes in everyone, including autistic individuals. But the reason I wasn't looking at it in this study is because that's where most of the research has been conducted today, the vast majority. So I was particularly interested in the eating side. We do have that, we have basic information on physical activity and exercise that we will be including in other models going forward to understand, they're separable, they're unique and combined. Because of course it's incredibly important. It's not an either or. You want to increase physical activity and exercise and address any kind of eating related concerns. Someone's asking if there are weight loss programs for adults with ASD with overeating when exercise is not consistent or helping in weight loss. I don't know of any specifically for folks with ASD. I know there are programs for adults with more broadly with developmental disabilities. And it's a little unclear because I don't think there have been many clinical trials at all to understand if how autism might interact with the ability of standard weight loss programs. So in other words, do issues around sensory related difficulties or divergent sensory experiences or cognitive and behavioral inflexibility, does that make a standard weight loss program more difficult, less effective for an autistic individual than for a neurotypical individual? And we don't have the answer to that as far as I know. So it's something that we, I'm hoping that research will address coming up. Someone's asking if we plan on creating a study that looks more at cause and effect, and the answer is yes, because we need longitudinal data. So we plan on conducting longitudinal studies to see over time, you can start to see directionality. Do we actually see that eating in the absence of hunger predicts some health-related outcome, and or vice versa? Does the health-related outcome or does the health-related condition predict eating in the absence of hunger? So you can look at it bidirectionally when you have more than one time point of data, whereas all the data I've described here is one point in time or cross-sectional. Sorry, I am. Okay, someone's mentioning that their child doesn't want more than one food on their plate, just one at a time. So it's it looks like sequential types eating. That is hopefully that allows you to present, the foods that maybe are less desirable at the beginning and then work your way to the more desirable food. Although obviously that would make it makes mealtime very long and could be arduous, I'm guessing. One person asked why there were only males in the gustatory mapping fMRI task. That was a convenience thing at the time. That would not happen if the study was conducted today. That study, it's small, but it requires a lot of time from the participant. And at the time, the research participants in that study or the registry of potential participants was almost all men, just because of where they were recruited from, but that's no longer the case. So if we were doing that study today, it would actually probably be 50-50 in terms of sex assigned at birth. Someone asked about the work on the relation of ASD and anorexia. And there is quite a bit of work on ASD and anorexia. And there's been some, let's see. So there's been quite a bit of work. It's hard for me I can't cover all of it here, obviously. But there's been some interesting parallels noted. So you often get people with autism on the ARFID side, right? When I said ARFID, the avoidant restrictive food intake disorder, you hear one piece of that, which is restrictive eating. And restrictive eating is characteristic of both ARFID and anorexia. However, the reasons behind that and other characteristics are very different between them. So ARFID has the restrictive eating, and you have the sensory stuff, and you have the nutrition and growth and the psychosocial functioning. On the anorexia side, you have all these issues around body image. And in both anorexia and autism, you have a great deal of cognitive and behavioral inflexibility. So people have often looked at, even more than in autism, looked at anorexia, they've looked at autistic traits or features in people with anorexia. And they see elevated autistic traits and features, particularly this cognitive and behavioral inflexibility. There's some pretty strong suspicion. A lot of this work has been done in the UK. They're documenting increased rates of anorexia in autism, and particularly autistic women and girls. But I don't have a lot more to say beyond that, just that we're in early stages for that kind of research. Are we running out of time, Janice?
We are. We're running out of time. But if you want to do one or two more questions, if you have time, that'd be great. And then we can wrap it up.
Okay. So someone asks about investigations of other senses, and I'd say particularly on the brain-based side. I'll just end there just for fun, since that's where we ended the talk, which was on the sensory side related to. It's a great question of why we didn't look at other senses, because we can only do one at a time, particularly given what I told you earlier, which was when we looked at the sensory-related factors predicting selective eating in autism, it was oral texture. So we'd love to do that. Of course, there's all kinds of constraints, including getting funding for such a thing. But the part, I'll just say this because it's a bizarre thing to study. When you actually study that in like the environment with fMRI, the way I've seen oral texture studied is using viscosity. What that means is you go from like a really liquidy, watery type thing to something that's really oily and thick. I personally don't think that represents what a lot of autistic people say about their experience of oral texture difficulties. So it's finding the right kind of stimulus that you can present in, let's say, the MRI environment to test it rigorously and look for brain-based differences. So it's a great question, and that's part of the reason we haven't done it.